Usage of UV-Vis in the laboratory. The energy needed to move an electron from one orbital to another is equal to energies of light in the visible and ultraviolet portions of the electromagnetic spectrum. When an electron moves from a higher energy orbital to a lower energy orbital, the difference in energy between the two orbitals must be released. One of the ways that the molecule may release this energy is by emitting light with an energy equal to the energy difference between the two orbitals. So, we can recall that Bohr used this concept to develop his model of the atom. So, in order for for an electron to move from a lower to a higher energy orbital, it must gain energy, and the molecule may get this energy by absorbing light with that specific energy content. So, since most molecules have energy gaps between orbitals that fall in the visible or ultraviolet regions of the spectrum, so studying by the way, the meaning of UV-Vis is ultraviolet visible spectroscopy. So, by studying the UV or the ultraviolet visible spectra of a molecule provides information on the characteristic energy gaps between molecular orbitals. So, this information in turn can be used to help to identify the molecule. So, the human eye is able to distinguish among wavelength in the visible region. So, kung lahat ng visible colors, si red, orange, yellow, green, blue, si indigo, at saka si violet are present, so yung makikita natin is white. So, if any color is missing, the eye sees that colors complement. So, the mixture of colors or complement or the mixture of colors that are left. So, maaalala natin, no, yung sa uh, color wheel. So, the colors that directly oppose or the colors directly opposite each other are called complements. So for example, si red at saka si green are complements kasi 'di ba magkabaliktaran sila doon or magkaharap sa opposite sila sa ating color wheel. So pag missing si red sa mixture ng ating visible light, ang makikita ng mata natin ay si green. So this means that the green-colored compounds appear that way because red is missing from the light they reflect. So in other words, they must absorb a light in the red region. So a compound that absorbs light around 470 nanometer is absorbing blue light and will appear orange to orange-yellow to the human eye. So, what is UV-Vis? So, UV-Vis, or the ultraviolet visible spectroscopy, it refers to the absorption spectroscopy or reflectance spectroscopy in part of the ultraviolet and full adjacent visible regions of the electromagnetic spectrum. This means it is a quantitative technique that measures how much light is absorbed by the chemical substance in the visible and adjacent ranges. The UV-Vis spectroscopy is based on the selective absorption of electromagnetic radiation in the 180 to 780 nanometer wavelength range. So the UV-Vis radiation has sufficient energy to cause transitions in bonding electrons as opposed to atomic inertial or the valence electrons. And thus, it is correlated best with the behavior of bonds and functional groups in the analyte. So the functional groups having electrons with relatively low excitation energies absorb strongly in the UV-Vis region and therefore called chromophores. So, pero kung meron tayong UV-Vis na UV visible spectroscopy, meron din tayong tinatawag na UV visible spectrophotometers. So, what is spectrophotometers? It is made up of stable source of radiant energy A transparent sample container, a device for isolating specific wavelength, a radiation detector which converts transmitted radiation to a usable signal, and a signal processor and readout. So each of these components will be discussed in reference to the carry 1-E UV-Vis spectrophotometer. So Sicari 1-E UV-Vis spectrophotometer will be our source of example in this presentation. So what is the principle of the UV-Vis? So this spectroscopy is based on the interaction between light and matter. So it is based on the absorption of ultraviolet light or the visible light by the chemical compounds that will result in the production of distinct spectra. So the amount of light that any solution will absorb is proportional to the concentration of that solution. So this is expressed mathematically as yung tinatawag natin na Beer's law. So the formula is capital letter A equals A, B, C, or A times B times C. So the capital letter A means the absorbance of the solution. The small letter A is the absorptivity of the constant. The B is the thickness of the sample, or of course the width of the cuvette or cuvette. Then the C is the concentration of the solution. The concentration of any solution can be calculated from its absorbance if we know the values of A, small letter A, and B. So the value of B is normally 1.00 centimeter. So, if we don't know the value of this small letter A, we can find the concentration of an unknown solution by comparing its absorbance to that of solutions with known concentrations. So, typically, we will prepare three or more solutions of known concentrations if we're gonna do the experiment. So, then we'll measure the absorbance of each and plot the calibration curve of the absorbance versus the concentration, such as uh, the one will be that will be shown here in our PowerPoint. So after measuring the absorbance of the unknown solution, its concentration can be read from the graph. So for example, if the solution of a known concentration had an absorbance of 0.60, we would consult the calibration curve and find a concentration of 0.10 m. So the sources. So a radiation source for spectroscopy must generate a beam with sufficient power, wavelength range, and stability for detectable and reproducible results. So many UV-Vis spectrophotometers such as the Carry 1-E use a deuterium lamp for the UV range and switch to a tungsten filament lamp at 350 nanometer for the visible range. The electrical excitation of the deuterium lamp at low pressure results in a continuous spectrum of emitted radiation from 160 nanometer to the beginning of the visible or the 375 nanometer. So then, an arc is formed between a heated oxide coated filament and a metal electrode. So when about 40 volts is applied to the heated filament, a direct current is produced, resulting in an intense ball of radiation. So then, at around 350 nanometer, the carry 1-E switches its radiation source to a tungsten filament. So the radiant energy emitted from a heated tungsten filament approaches that of a black body, and thus it is temperature dependent. So the operating temperature is usually 2870 um, Kelvin, which results in radiation of the 350 to 2500 nanometer range. So the lower limit is due to the absorption at 350 nanometer by the glass container housing the filament. So, of course, most of the experiments, we will encounter some problems. But uh, the problem are the problem of source stability can be resolved by using a dual beam system where radiation is directed at the sample and the solvent reference at the same time. So then the fluctuations in the source will be cancelled. So the carry 1-E utilizes a dual beam system. The sample containers that will be used in the experiment. So the sample containers are the cells or cuvettes. Cuvettes must be constructed of a material that is transparent to radiation in the wavelength range of interest. So containers of quartz or fused silica are necessary in the UV range and can be used into 700 to 3000 nanometer infrared region. So the glass and plastic can be used also in the visible region as well. So to minimize reflection, caveats should have windows that are perfectly normal to the direction of the beam. So quartz caveats with a 1 centimeter path length were used with the Carry 1-E. So next we have the wavelength selector, radiation detector, and signal processor. So shown here in our diagram is the schematic Carry 1-E. So as we can see here, the number one part is, or the number one part is called the D2 Q QI lamp. So it is the radiation source. The grating one is the colored glass filter wheel that isolates a rough region of wavelengths around the desired wavelength. Then the parts of the um, wavelength selector, radiation detector, and signal processor is the entrance slit. It isolates a thin beam of radiation coming from the filter wheel and limits the range of incidence angles. The grating 2 is a holographic grating that disperses the radiation, allowing a very precise selection of wavelengths. So the grating is 30 times 30 by 35 millimeter in size and can move between wavelengths at 3000 nanometer per minute. The exit slit is the radiation from the holographic grating is reflected by a mirror to the exit slit. The desired wavelength is selected by rotating the grating relative to the exit slit. So then the radiation of undesired wavelengths is absorbed by black matte walls surrounding the grating. The chopper 1, the radiation from the exit slit is directed by mirrors at the first chopper. So the chopper consists of three sections as shown in here in this figure, and it rotates at 30 hertz. So the chopper has a cutout section as shown, which allows radiation to pass through the chopper to a mirror which directs it to the sample cell. Then the next section on the chopper uh, is a mirror which reflects the beam to another mirror and then to the reference solvent cell. So the light that hits the black section is absorbed. Thus, the beam hits the sample and reference almost instantaneously. Next, from chapter 1, we'll go on with the sample position. So its function is the beam is focused to the center of the sample compartment to allow maximum light throughput and reduce noise. So chopper 2 or the second chopper, it ensures that the sample beam and reference beam hit the phototube detector at the same position and angle since the detector is sensitive to these quantities. So the mirrored section reflects the sample beam to another mirror and then to the phototube detector. So the chopper then rotates to the cutout section, which allows the reference beam through to the detector. The solid section of the chopper absorbs all radiation and blocks the beam from hitting the phototube detector. So the last part, which is the phototube. So the phototube detector first corrects for any residual signal in the detector by zeroing itself during the moment when the black section of the choppers block the light beam. So next, the sample and reference beams are sequentially measured. So the transmission is determined by T equals sample, close open parenthesis, sample minus dark signal divided by the close open parenthesis reference minus dark signal. So the transmission signal is sent to a computer where it is converted to absorbance. The result is shown on the CRT. So, in the experiments, there will be some interferences that may cause errors, or that will be the cause of errors in the laboratory. So, the most obvious source of error in correlating UV-Vis absorption with DOC is from the non-absorbing organic material present in the sample. So, simple sugars like aliphatic acids, alcohols, and amino acids such as glycine do not absorb in the UV range, which results in underestimation of the DOC. However, these compounds are those most likely to be consumed by microbes as food and are not expected to be present in natural water samples from rivers and lakes at high concentrations. So there are also other possible sources of interferences with the desired UV-Vis spectra. So that can come from the particulate matter like the nitrate or the nitrite, chlorine, and or the ozone in the sample. So the nitrite are a nutrient ubiquitous to natural water. So the nitrite it absorbs at 330 nanometer due to a uh, N to pi transition. So nitrite also absorbs at 280 nanometer and 360 nanometer. So, but it is not usually found in natural water or treated wastewater. So the nitrate used to lower the pH of our samples may have interfered with the absorption spectra by giving a higher absorbance than for sample alone. So the presence of the oxidants, such as chlorine or ozone in the samples can cause a reduction in the absorbance at 254 nanometer without the corresponding reduction in the OC. So these small amounts of those compounds can partially oxidize some of the NOM to carbon dioxide. So in the process, less aromatic and absorbing byproducts are formed. So, for example, the example of these the byproducts of fluorine, addition to natural water, can result in a di and trichloroacetic acid, dichloroacetic acid, and trichloroacetic acid. So it also has been shown in the laboratory experiments that ozone addition to fulvic acid solutions causes a reduction in absorbance at 254 nanometer. So the particulate matter can affect the UV-Vis absorbance by scattering the incoming radiation. So this scattering by suspended material in the sample increases with particle size with maximum scattering for a given weight of sample occurring at a particle size equal to the wavelength of incoming radiation. Therefore, the UV radiation is more sensitive to turbidity as it is scattered by smaller particles than its radiation of longer wavelength. So although the natural water sample in this study was unfiltered or there is a little particulate organic carbon that is present and no particulate material was visible, uh, Taubes et al. Found that unfiltered clear samples of natural water were adequate to perform error-free concentration studies with UV-Vis absorbance. So then, let's move on to the calibration procedure. So the calibration procedure, it's standard curve relating to A 254, so the concentration of KHP and ppm, or in ppm or parts per million C, was plotted as shown here in this figure. So a typical regression analysis was performed on the data, resulting in the linear curve fit and equations as shown. The relevant parameters are displayed in the results section, so all errors are reported at the 95% confidence level. So the result that I will be showing here is just one of the results from the experiment. So the absorption spectra from 200 to 700 nanometer for the samples of glutamic acid, salicylic acid, tap water, soil extracts, and wetland water. It's from the wetland water from the original pH and pH2. So here, shown in the figure, the Prado wetland spectra. So we can see that the acid or the acidic wetland has a higher or have a higher absorbance than the wetland alone or than the acidic tap water. So that is because, based from what I have told you earlier, the presence of some oxidants or may interfere with the results of the absorption of our given sample alone. So the acid wetland is, of course, from the 200 or 200 to 700 nanometer, is that the acidic wetlands has a point 17, or estimatedly, sakto ba kung term? With an estimation of point 0.17 absorption. So there are also other results, but since this is just an example, so that would be all. And I hope you have learned something from this discussion.